We'll start the session in a few minutes. By the speaker.
Okay, good morning, and I welcome you for the 7th day of this workshop. And I'm happy to introduce today's speaker, one of the main person in the organizing this event. That's Dr. Gautaman Namali. So he's a structural and computational biologist with more than a decade of experience in predictive modeling and biomolecular simulation projects. Dr. Anamali's skills involve molecular docking, molecular dynamic simulation, post-grain modeling, machine learning, data mining, data analytics, and molecular modeling. He has obtained his doctoral degree from Jawaharlal Nehru University, New Delhi. So he did his postdoctoral research at Iowa State University, University of Wisconsin-Madison, and Rice University in USA. So currently he's working as a assistant professor in Shastra University. And he has contributed to multiple diverse projects that focuses on basic and applied sciences. His experience in computational structural biology is critical in finding answers to the bigger questions in life. So I welcome Dr. Gautaman to take.
Thank you, sir. Thank you, UV sir. Thank you to the Shastra University for giving this opportunity to organize this 10-day workshop, and to Accelerate Vigyan and DST for supporting us. So today, what we are going to do is we have 4 sessions. First will be a lecture, followed by a hands-on, which will be conducted by myself and Dr. Krishnakant Gupta. Then followed by session 3. After lunch, we will have again another lecture, and it will be it will be a hands-on followed after that. So today, the first session, what we're going to see is the topic given to me is about biological networks. Most of the stuff related to biological networks has been covered a little bit by Professor Ram Swami from IIT Delhi yesterday, and a little bit in detail by Dr. Satya, and also some basic stuff that you all would be familiar about with the graph theory part, which was conducted by Dr. Krishnakant. And then finally, one of the application that that you all were familiar with or got familiarized with was by Dr. Ashok Parnepan yesterday in the last session of the day. So without further delay, let me start sharing my screen. Okay. Okay. Is everyone able to see my full screen, the slideshow? Okay. Okay, yeah. I understand. Okay. Um, so you're able to see my screen, right? Okay. Sounds good. Um, so just a minute. I know I asked you a question and you guys probably have not enabled your unmute button. So we'll work on that. Meantime, if you have any questions, do interrupt, and then I'll try my best to answer your questions. So the first thing that comes to mind when you talk about biological networks. Now, most of the time, we are, as a biologist, we are familiar with biological networks, a visual representation of how a system looks like. And this is like something that's the bread and butter of biochemistry, the pathways, the molecular metabolic reactions. All these are in one way or the other networks. You're unmuted yourself. Mute yourself. Okay. Uh, no, why?
It's mute only.
Are you able to see my screen?
Yeah.
Okay. Are you able to see the next screen?
It should be modeled. It's right.
You're able to see it?
Yeah.
We're able to see it. Just give us a minute. There is some network issue. Live stream le பாருங்க. சௌந்தர்யா. உங்க live stream ல என்ன தெரியும்? Automatic panelist host பண்ணிருந்தா. Okay. Okay, you become the host now. Yeah, you become the host. Is it come back for you?
मोबाइल से कैसे करूँगा, यहाँ तो नेटवर्क भी नहीं है।
Oh, really? Vodafone or Airtel?
मतलब BSNL है, BSNL का इतना है।
BSNL is what is down.
And जियो का भी नहीं है उतना।
Yeah, BSNL fully down. Live? Can you refresh the page? Try refreshing the page. Because it says here live on custom live streaming service. Yeah. Yeah. Okay. Okay. Murphy's law. If something has to go bad, it goes bad. Okay, so we'll make up time. I hope that they are allowed to talk. Okay, you will be allowed to talk in a few seconds. As I said, interrupt at any point of time. Now, carrying on from where I stopped earlier, the first thing for um, any issue or any project or anything that you want to start with systems biology, you want to ask what can or what should be modeled. Now, this is most important. The reason being that until unless the question is clear and you have the data for answering the question, you will not be able to find the solution that is required. It will be GIGO, so which means garbage in, garbage out. So you have to be very clear about what you want to model and what you want to do with the model and what is the answer that the model going to give you. So it becomes kind of like from a biologist point of view, when you're looking at an experimentalist for whom the output is known but then not known to a large extent, this becomes a very clear case of you have a clear input, you have a clear output, you want to know how what is happening in the middle. That's not how biologists look at systems, look at many of the problems. So for them, some of the outputs are not so very clear. So you might think that then in this case, when we know clearly what we are looking for, then how does this help me? Or what am I really going to do with the systems biology or a project that I want to involve systems biology? Now, again, it's a circular argument. You need to know what you're going to do. So the question could be, what is happening with the reaction, or how long does the reaction take, or what is happening in the few microseconds that once the reaction has started, or how does the pathway behave when there is an external stimuli or an external perturbation happening. These kind of questions are what most papers that talk about systems biology answer, and that has been in the vast literature all through. So when you look at the word biology in systems is not just randomly put there. It's because you are actually trying to model something that is a living entity. Now, a living entity can have a nucleic acid, either DNA or RNA. It shows some level of self-organization, in terms of organelles, in terms of the genome structure, and has evolved over time. So there is a time component involved in it. So if we break down a particular living entity into one of the consisting of these three criteria, then the goal is not to imitate life, but to understand the universal logic and properties of the living systems. So this is what is the main big picture of why you want to do systems biology. And in case if your question or if your big picture is something other than this, then probably systems biology is not the solution. It might be something else. You can still do a computational modeling, a predictive modeling, but it need not be called as a systems biology. Um, Now, the first and foremost thing that the need for systems biology is because for 200 years or so, we all have, as humans, we all have studied in the reductionist point of view, meaning that we reduce the big component of the cell and then break it down into smaller fragments. And as we go down, we narrow it down to individual molecules or individual entities that interact with each other, that regulate each other. What you have to understand is that there is a huge interconnective crosstalk that happens between different processes, different components such as transcription, translation, post-translational modifications, and this happens all throughout at any point of time in the cell. The cell is a very crowded environment. Now, if someone asks you what the definition of systems biology is, because right now I'm being very vague, and if someone asks you like, okay, you attended 2 days of workshop that covered systems modeling and network modeling, tell me what is systems biology? So systems biology, you can say that maybe as defined by the NIH USA, is an approach in biomedical research to understand the large picture, be it at the level of organism, tissue, or cell. Now, you might think that the large picture compared to an organism to a cell, the organism is at a much higher hierarchy. But even within different hierarchical levels of the living entities, you can have understanding at the a large level. Even a cell is a large enough complex entity. So we do this by putting the pieces together, and it is a huge difference from the reductionist biology where you take the pieces apart and then you see how it is fitting together. So most of you might be familiar with how small boys, they when you give them a toy like a car or some kind of toy, the first thing they would want to do is break it down into pieces and see how it's working. This is very familiar if you have a sibling uh, who's a male, uh, and the first and foremost complaint is like, darn it, it didn't survive even 5 minutes of its life. But that is actually what has been driving humans for many centuries. We know that system as such, even with the level of cell, is complex, and the most easiest way to understand any complex thing, and this is a very basic instinct, is that to break it down into smaller pieces that can be understandable, or to put it in quotes, chewable, so that you digest the information, which is what even this workshop is all about. All right, like we can have a high-end workshop for just one day. We can concentrate it and give it like 4 sessions of the entire computational biology and data science, but then you wouldn't understand. It will be a very hard to digest. So we have divided it in 10 days with 5 teams and so on. So this is like a basic thing that has been driving us humans for a very long period of time. So you start with computational part. So where do you begin with? You need to have a model, or you need to have a skeletal structure of the model that you want to work with. You add some cell biology part, which is basically from the experimental data. Then the omics data that you get from proteomics, from metabolomics, and then the transcriptomics. Then you know the genes that are involved in the which give rise to the gene product. And in eukaryotic system, there are isoforms which complicate the system further. So you need to know what are the splicing events that are happening, and then of course in human systems, you want to add little bit of immunology as well. So it's a kind of like a mix, and it has various components added to it. Not everything is known about every component, but we try to do certain things by starting from somewhere and then keep refining it as we go on. And that is to some extent a trial and error method. And this is what I was just mentioning, that for more than many years, we have characterized many of the physiological and biochemical mechanisms using a reductionist approach. And this actually is very useful. Why? Because when you are trying to put an entire pathway into context and you're trying to simulate a pathway, you're trying to see the dynamics of a pathway, you still need to know the individual components of the pathway, their interactions, their dynamics, and so on and so forth. So whatever has happened in the last 2 centuries since modern biology started, and with the advent of omics and everything, all that information is still required for us to come up with a systems level understanding of uh, this entire whatever model you're trying to do. So then comes the question, does biology have deterministic laws and rules? Yes, it does. It still follows to a large extent the laws that are defined by physics and chemistry, when it comes to molecules, when it comes to DNA binding, when it comes to transcription factor binding to the promoter region. Everything is pretty much deterministic, but at the same time, the outcome of those deterministic events are not deterministic, but they are to a large extent stochastic, and it depends on many multiple variables. So if we go by the logic that, okay, I know that systems biology involves 5 components, or for the model I want to work has 5 components, let me put all the 5 together and then now I got a model. Well, that's the same analogy that you can give by saying, let's say, I want to make grape juice, which basically you reduce it to having carbonic acid and alcohol. Uh, I have kind of sanitized it. Let's say you want to have a cup of wine, you can't just add alcohol and carbonic acid and say, okay, that's wine. It's not yeah, okay, got back. Okay, so um, so something, the reductionist approach is not unique to biology. It's more something that is, as I said, it's a very basic human tendency, and it traces back to even to like Julius Caesar, where divide and conquer, and in India the British Raj. We don't have to elaborate further. This is a very common approach if you want to understand a very complex entity and you want to break it down and see how it works. So it has its own limitations. As I said, with the example of the grape juice or wine, you can make a cake. A cake contains milk, flour, eggs, butter, sugar, and candles. But if you mix all of them, that's not a cake. That's just a mixture. So textbook concepts, sorry, are fragmented, and this is where we tend to, and this is how most of the teaching in biology has been happening over the last couple of decades, including India. So we teach each component separately, like microbiology separately, biochemistry separately, immunology separately. So you compartmentalize all these concepts, but there is little understanding of how all these different components come together, and there is an overall design that's involved in it. So from an engineering point of view, systems biology makes a lot of sense, because it is, you're not considering the biology as an entity, but as a circuit. So you have different components of the circuit. You have the flow of information happening from one part of the circuit to the other part of the circuit, and you're trying to understand how this circuit runs, what could perturb the circuit, and how this circuit could be made even better to give the desired output. So the non-linearity component of the entire system is what adds up to a great level of intrigue. Okay, Aaron Kachalski in 1970 said that biologists can be divided into two classes: experimentalists who observe things that cannot be explained, and theoreticians who explain things that cannot be observed. So for this makes a lot of sense, because experimentalists try to uh, recreate something that they have observed in nature, in real life, and then try to understand what is happening in it. But there are a lot of unknowns. Now, a theoretician, in contrast, or a computational person, in contrast, knows exactly how the model would look like. But unfortunately, such a model, finding a model like that may not be so easy, because that may be very far away from reality. Then in case, how do you bridge this? How do you bridge these two entities into one common concept? So Reconstructionism took off sometime in the 80s and has been driving. So to dispel the myth that systems biology is only myocardial interactions, a lot of research has took the entire heart as an entity, and then you actually see that you actually see whether you are able to uh, mo- model the rhythm of the heart, and the waves of how the heartbeats are happening. All this has been happening for like a lot of studies have been happened on that, and all those do come under the umbrella term of systems biology. So the emerging paradigm that proposes in contrast to reductionism is to put the isolated parts together again and analyze the interactions within them and the environment that they are playing. So this needs well-controlled in vitro experiments, which we cannot do without, and it needs mechanistic rate laws as well. So these are deterministic in origin. For any system, you want to have a valid means of a problem that is simplified, a convenient terminology, and a convenient mathematical representation of the system. Now, this simplification is again, this is what Ram sir touched upon yesterday. DNA model, someone can say, let us consider DNA as the uppercase D, or someone could say that let us look at this two curved lines as DNA, or someone could go into each atom of the DNA, just like how Watson and Crick built the DNA model, and you can consider that as the DNA model. Now, with each of these three uh, models that I just mentioned, one is just a letter, the other one is an abstract double helical structure, the other one is an atomistic double helical structure. Each of them, the question is, are they enough to explain what DNA is? Yes and no. There are advantages to each of them. There is a level of abstraction that has happened to each of them. At the atomistic model, you might think, oh, it's very fine grained that I know what DNA is all about. I can see all the atoms. But do we know how the DNA winds across a histone? As Professor uh, uh, we had a speaker who talked about epigenome, epigenetics, Vijayalakshmi ma'am. So, do we know by just by looking at the atomic structure? We don't. Okay, so even when you look at different models of DNA, it is not enough to explain everything about the DNA. Okay, so that is so the level of simplification or level of abstraction you want is all depending on what you are trying to achieve. Um, in a biochemical equation, the laws of particle physics do involve, and that is, it still happens. But what do we do when we write a chemical reaction? We tend to ignore the laws of particle physics. We do not look at that because it's happening. we assume yes, it's happening on time and on schedule, but we do not think that as a variable to be incorporated into the model to define a particular system. And this is the same thing like where you are simplifying a particular entity by replacing the complexity of it at submolecular levels with some average behavior. Like you might have 5% of the pathways not behaving in the same way, 5% of the cells in a colony not having, not being the same way, but still the average overall behavior, a generalized behavior is still remaining the same. So one of the example is that you can have biochemical interactions that happen between the hormone insulin in relation to the glucose levels in a diabetic patient and to the level of adrenaline that is present because of which this glucose is being produced. So when you look at this kind of a system that has these components, then the question is, do you want to study what is happening as a result of a high concentration of adrenaline and the yield of glucose, or do you want to see the mortality rate of people who have an insulin resistance? I know these were totally different questions, but then for both of the questions, you have these components that are simplified and you can answer them in a different way. Convenient terminology here means you do not uh, define something that is like, or you make up terms that is cosmetic in origin, meaning that it's you still follow the terminology that biochemistry gave us, the terminology that kinetic laws follow, and the mathematics and network biology follows. So some of the terminologies that is not because now it's a new field, you define new things, but it is basically borrowed items from different areas that actually have a generalized or a common understanding between the communities. Now, coming to the mathematical representation, here we always look at the model and we define everything in terms of uh, uh, in many different ways. And this is where our school knowledge of mathematics finds a clash in when you understand systems biology. In most of the time, I can tell from my experience, and I'm pretty sure it's the same for you. In your maths class, in your school, the teacher says that if you miss one step in the derivation, you get the wrong answer. So either you get 10 out of 10 for that question or 0. So it becomes a binary concept at that point of time. But when you look at systems and when you define them in terms of mathematical equations, mathematical, either it's ordinary differential equation or partial differential equations, it's not binary. It depends on what you're asking and where the answer is going to be and how you have defined those equations. So this is another thing that you have to understand, that in systems biology, you do not have a binary outcome. It's not yes or no. It's not qualitative. It's quantitative, and at also at the level of why certain things are happening the way it's probably happening. So I'll skip all this. So example is that in a biochemical model, systems responses can be explained via kinetic parameters. And in a cell population model, growth characteristics can be explained via cell cycle phenomena. So these all do not have a binary outcome. There is a range of outcomes that can be expected from them. Most important question, why modeling is necessary and why do you have to make a model? It is, as I said, it's an abstract representation. Even if you draw something on a piece of paper, that is a model. But in the case of systems, the word systems in biology is because you're using computation, and so it's not just human readable, it's also computer readable. It's supposed to be computer readable, right? Um, there is a huge amount of complexity because of which modeling tools is necessary, and understanding this complexity increases the study that what type of experiment can be done or can be designed to extract the information. I'm probably running out of time, so I'm going to skip to this main part. Okay, so this is the system, and you might think probably I didn't have my morning coffee and I'm calling this as a system. Um, this is showing a reaction where D-ribose 5-phosphate is getting converted to beta 5-phosphoribosyl alamine with the involvement of ATP and glutamate. You might say this is a chemical reaction. Yes. But at the same time, I know something more about what is happening rather than just telling the names of the entities. I know what is happening to the phosphate group. I know what entity or amide bond is being formed, and so on and so forth. The same thing can be explained like this. And here I have few more information added, where with the use of bold arrows and thin arrows, I can say what is the main part of the reaction, what is the other entities that are involved in the reaction, and the other important part is the feedback. So if ATP concentration increases, then there is a negative feedback that's happening at the initial part of the reaction. So this also is a model. This also is something that can be defined. So when you look at the different stages of where you start from a system or a model, which is seen in real life, to a mathematical representation, we go from different parts, and you can stop at any one part depending on how much information you have, how much data you have. You start with the biological system like a colony of cells, a colony of bacterial cells. You make a mental model, which is nothing but what you do on a pen and paper. Most biologists are familiar with it. And then you make a scheme of the model where, like for example, the protein X activates the production of protein Y. And you make a process model after that where you determine what are the rate constants, what are the parameters, what is the activation required for a particular component to be converted or used up or being produced, and a dynamical model where you have the denominator of time involved to understand what is happening. And when you plot the results, or when you plot the data that is an outcome of the dynamical model, you get a quantitative result. So this is what all it is about, and this can be applied to any entity that you want to study. This is very important, and in case this might be a repeat for those who know about systems biology. Um, Most of the time, when you are discussing your work, you say, okay, we did a systems biology approach, we did blah, blah, blah. The most important question someone would ask you is, what's the scope of your model? uh, it's probably nothing, but what is your model describing? That's what scope is. It describes the properties of the biological system. When you construct models where the properties are not compromising the basic results, then you incorporate them. Otherwise you tend to limit them out of from your model. So what are the model elements? How many species does it have? What are the equations? Is it a partial differential equation? Is it an ordinary differential equation? What are the constraints? What are the K max values, K I values, K A values? All these things is what? Everything. The entire component or the entire list of things that you have put inside to make a model is what scope is. The different types of model: static, dynamic, deterministic, stochastic. Out of scope for this particular talk, but I just letting you there. I don't have to tell you this. What is the advantages of computational modeling? It's very easy. The one thing that I want to stress upon is time and space can be stretched and compressed in any model, and the reason is uh, it's because of this. Some of you might have seen this book or the website called Biology by the Numbers. Um, here you have different entities of this cell and the time taken in terms and the units that are there. What you see here is that not everything happens at the same time. Not everything is happening at the same scale. Something happens at the level of hours, like the protein's lifetime, and something happens at the level of minutes. Something happens at the level of microseconds. Okay, there are some reactions. If the free energy is like more negative, then it happens in a fraction of a second. It's very spontaneous. Reactions are spontaneous. So when you have a model, the biggest advantage of a systems biology model is that you can make the model have different time scales even within one single model. Okay, so certain events can be at the microsecond level, certain events can be at the nanosecond level, certain events can be at the second level, and they all go in synergy with together. This is the biggest thing, because a cell, like developing cell, a cell that's in Drosophila or in C. elegans, the time scale of the entire cell developing into something else is at the level of hours. But in the case of the reactions that are happening because of which the development happens is at the level of seconds. So you tend to you can stretch, you can compress time in a model that you build computationally. Um, as Ram sir also said, you can't model everything. You have to know where the limit is, and identifying the limit is also a bit of a trial and error method. So it's neither recommended nor practical to include all aspects, because the if you have too many parameters, then you end up making the system very complex to even compute and understand, and you do not know what is happening. Uh, and the other challenge is that many of the parameters that are involved when you're defining something is not clear. You do not know because we do not have data. Those experiments either cannot be done or cannot be available because of the instrumentation. There are so many technical challenges also. So this is something that Dr. Krishnakant covered yesterday, biological networks. I'm not going to go into detail, but the word network here indicates that many of the biological entities can be defined as a graph. So these are not just pretty pictures. Understand that those who come from purely biology background, it also is machine readable, and you can define, you can construct the entire, do a lot of calculations based on a pictorial representation of the system as well. So there are different types of biological networks: transcription or gene regulation networks, protein-protein interaction networks, signal transduction networks, metabolic networks, correlation networks, neuronal networks, phylogenetic networks, and ecological networks. Most of you who are from the biotech side, involved in computational biotech, might be limiting yourself up to the first 5 parts of the networks, but you still need to be familiarized with what other types of networks are there. Okay, so how one gene is controlled by another gene product, very familiar to all of you. I think you all know this. I don't have to go into detail. So this is a gene regulatory network. The arrows indicate what is activating something else, what is inhibiting something else, what is being regulated, whether it's a positive effect or a negative effect. There's a lot of information, layers of information involved here. Protein-protein interaction network is like very common these days. If you remember Dr. Emily Steen's slide of the crowded cell, the proteins in the cell are constantly talking with each other, bombarding with each other, bumping into each other. So whether the interaction is transient or it remains for a long time indicates how the proteins are interacting. What I want to emphasize is that when you look at a pathway, this is a activation of the ERB, ERBB kinases pathway. This is not in the pathway way usually you are familiar with. But what you see is each color represents a particular protein that's involved in the pathway, and that these are all the different conformations that's available, obtained by experiments through X-ray or NMR. And what you see here is that the way they interact with each other, they have a specific shape complementarity, and if you stack the entire all the proteins that are involved in the pathway close by to each other, you can see that anything that's happening to this particular protein is going to have a very quick effect on this particular protein. It's not very far away. Okay. This is a CDC global protein kinase phosphate interaction in yeast, published in 2010. The thickness of the lines, the color of the lines indicate the different types of interaction that the proteins are having with each other. So kinase and phosphatases go hand in hand. They are opposite reactions. So then the other type is signal transduction networks. This is the classic fight or flight, based on an event, a stimuli. Either you fight it, like the boy, he's kicking the doll, a creepy doll in the middle of the hallway in the night, or you can run away from that. Someone is asking, sir, for modeling a system, we refer multiple papers, but different papers would have followed different experiments. Yes. So what you do is, when you look at a particular system, the first thing you need to know is whether the experiments that have been conducted, what level of reliability do they have. So most often what we do is that we collect different information from different papers, and then we see, let's say for the same entity, like say kinase. Okay, there might be 2 papers or 3 papers, 3 literature evidences that talk about uh, whether a particular talk about different concentrations of the kinases. Okay, and different areas in which the kinases can phosphorylate some other substrate. In which case, you are it's very well okay to begin with all 3 different values, but when you finalize the model, when you refine the model, you have to stick with one value or one particular set of conditions. These set of conditions may not origin from one single literature. It might be from multiple literatures, and uh, you have to see whether those values make any sense when you run the time dependent dynamics. Okay, so it is general practice to club different data coming from different sources, but you pick and choose based on how the model is performing after the refinement of the model. So signal transduction networks. Okay. So signal transduction networks, I don't have to go in detail. There is a stimuli, there is a huge amplification, and then there is an outcome, and then coming back to its pre-stimuli state. Metabolic networks, this is signal transductions networks are a part of metabolic network, and you have databases like KEGG, EcoPsych, BioPsych. All these have huge metabolic pathway for different organisms, and they involve not just substrate product but secondary metabolites and other metabolites that are part of the entire metabolism of the organism. I'm reminded of metabolic network when I look at this image that I took on a hike in Himachal. So this spider cobweb is actually how a metabolic network would look like. Okay, so it's huge, it's clustered, there are too many interactions, and it's quite messy. The most common biological metabolic network is something like this. Here in this case, it is redesigned like a metro map, the metabolic network as a metro map, or like a railway map. So it makes easier for the humans to understand, and at the same time, this is also machine readable. Uh, this is a classic culprit in all cancers, and you can see that the P53 is not just involved in one particular part. It's involved in autophagy, it is involved in apoptosis, proliferation, and so on and so forth. The P53 network is itself a huge network. And if you want to study something about P53, taking a subset of this network itself is challenging enough because you don't know many things about that. Correlation networks are basically you calculate statistical correlations and then you see whether the node corresponds to a variable, and you find out association between two nodes, whether there's a positive correlation or a negative correlation. Like for example with this, so BioNetStat is a tool that with which you can do you can find correlations where the more darker the color it is, it's having a very high correlation, and the one with the pale color, a very correlation. Neuronal networks are something that the most common model that's used is C. elegans. It has a very few sets of neurons. So seeing how the different neurons make synaptic connections is something very interesting, and that's why it's a very good model organism for neuronal networks. And the neurons respond to a certain stimuli, and the neighboring neurons also get activated because of that. And so the post-synaptic potentials, the excitation potential, all these can be mapped and can be defined. This is a neuronal network where this particular part was damaged, and then what is happening after the damage and how the network gets recovered. So this is the very usual way of a neural network being studied. Then phylogenetic networks, however, these are not in terms of a typical graph that you are familiar with visually, but they are telling the different relationships between the different organisms and what is the evolutionary time point and how they have evolved over time. So when you look at this is the tree of life, phylogenetic network, and the red lines indicate to horizontal gene transfer that has happened between the various bacterial species, and how because of that one horizontal gene transfer event with a particular time involved, you can actually see the branch getting added up and expanding further. So even for this particular type of a network, you can do a time dependent simulation and analysis. Ecological networks is what most evolutionary and ecologists study. It's a very classic way of seeing the food chain or the food web that you are familiar from your school textbooks. But here you have two layers. One is an autotroph layer and a heterotroph layer. So something like this. So this is a tool called as EcoPath. EcoPath, it tells you the branch or like what is the level of the distance between one functional group to another, and how the flow of the energy or the food is going from the autotrophs to the various heterotrophs. So this, I'm not going to go into detail. How do you do analysis? This was covered very much in detail yesterday by Dr. Krishnakant and Dr. Satya, and you are all very familiar with it. So some of the global parameters that, let's say you have a network, you have built a model, you've connected all the nodes, you connected all the edges, you have all the connections, then what are the global parameters that you can look at and then make some sense out of it, okay? Degree centrality, average degree, average neighbor degree, eccentric centrality, shortest path, clustering coefficient. All these are fine and dandy, but most important thing that comes is, what is the scientific question? What are you trying to get from the network that you just built? If that does not make any sense, then probably you have to go back, go to the blackboard, and then do the entire thing again. The reason is this tends to I tend to philosophize here. Science is not like mountaineering. Just because there's a mountain, a virgin mountain that its peak has not been scaled, you don't put on your gear and then climb up the mountain and then reach the peak and then say, yes, you are the first person to climb that mountain. science is not like mountaineering. The reason is that just because something was we don't know about, or something we hasn't been explored, is not a good enough valid reason for you to take up that endeavor. So even if it's an off-beaten path, there are a lot of other things. It depends on how you ask question and what you are trying to get out of it, and that remains the central crux of anything in biology, and especially when it comes to network biology and systems biology. The scientific question is at the front and foremost. Everything around it is depending on the scientific question. If this is flawed and this is weak, your entire premise just falls flat. This is a computational or a computer, the SBGN standard, which is derived from KEGG, uh, where they use the tool called Vanguard to compute and display the various components of the metabolic pathway. What you see here, these different symbols, is almost similar to what you saw in Cell Designer. It's not just pictorial again. You can do a lot of calculations with that. There are different ways in which you can lay out visually a graph. Like for example, here it's a force-based layout algorithm, and it's a hierarchical uh, layout that's used. Both are the same number of nodes, same number of edges, but just that the visualization is different. It doesn't matter what it is. There are different ways in which you will encounter biological networks in literature. You have the like a network that's computed like this, or a network layout like this, or a network like this. What I showed you earlier, the metabolic reaction of ribulose 5-phosphate, is something very close to what this is. Okay, so that's still a model. Now comes the main part of the entire systems biology. Um, if you are going to embark on a biological network analysis, the first and foremost is defining the ordinary differential equations. Okay, these are very simple equations, very easy to construct, and they basically describe the change of the variable with respect to time. You can describe entire model, entire systems with given kinetics and parameters that you know. Like for example, a gene getting activated or having a positive autoregulation. One of the common things that for these kind of setups is the Hill function. Dr. Satya touched upon it yesterday. Some of you might be familiar with this. For the definition, for the benefit of others, I'm just going to breeze through the Hill function. There are three components basically involved in a Hill function. One is the activation coefficient, the maximal expression level of the promoter, and the steepness of the input function. Okay. Now this n, the value of n, is what tells you how quickly does a gene get activated or quickly does a gene get repressed. The way this Hill function is depicted is through a step function. So the step function where n equals, you have a very steep change. It's a binary change from 0 to 1. But the more values you increase to n, you can see that the steepness changes. And this is described by the activation of x, activation of y by x. So at the half level of beta by 2, the half expression of the gene required to set off the activation is beta by 2. At that level, you have the steepness changing from a dormant to an activated form. So this Hill function is very useful when you are trying to model a single gene. Like for example, uh, you have um, positive autoregulation, negative autoregulation, which are involved in different transcription factor modeling. So this cup K is the activation coefficient, and it is in terms of concentration. And you have the beta value, which is the maximal promoter activity. So the Hill coefficient determines, as I said, that determines the steepness of the input function. The larger the n, the more step-like the input function is, what has been observed by Yuri Alon and his co-workers is that most of the biological reactions have a Hill coefficient up to 4, not more than that, and it tends to be within that value of 4. Again, in a negative autoregulation or a negative repression, the Hill function is basically the reverse or the inverse of what you saw. So the Hill function for a repressor and the Hill function for an activator is very pretty straightforward to derive. When you have a metabolic network, the first thing, as I said, now this is a schematic model. So from a mental model, we have gone to a schematic model of the first 3 steps of glycolysis here. What you do is you limit the system to what entities you're going to study, and you balance these equations by generating a stoichiometry matrix, and then you assign the kinetics. This is what the entire mathematical modeling of a metabolic network or a biological network comprises of. Okay, so to give you an example, this is from basic chemistry. All of you must be familiar with how you balanced your equations in chemistry with right hand side should be equal to the left hand side. The other way around of doing the same thing, but it does not involve some amount of the directionality, is by generating a stoichiometric matrix. So the stoichiometric matrix is nothing but if there are two entities, S1 and S2, that are giving rise to 2P, two entities of the product, then the stoichiometric coefficients of S1 and S2 are -1 and -1, and for P is 2. The sign here indicates to some extent what is being converted, what is being used, and what is being produced. So I'll just skip over that, and okay, I need to do this. Okay, sorry. So for each of the entities that are involved in a reaction, you can describe them using an ordinary differential equation where the rate of use of S1 over a period of time is by minus V, and that's the same for S2 because they both are involved in one reaction, V indicating the number of reactions. So V1 to Vn, and then the rate of formation of P is 2V. So here you can see that the coefficient here, of course, the stoichiometric coefficient of 1 is not written, it's assumed. But for those that have more than 1, then you explicitly write those. So these are the ODEs that you come up with. So for any equation, the stoichiometric matrix, I'll just skip all this. Of course, I'll share all these slides to you. You can go through them, and I'm going to be here the entire day, so you can ask me at any point of time about what I couldn't cover. But the stoichiometric matrix is nothing but for an equation like S1 plus S2 plus S3 involved in one V1 reaction giving S4 and 2 S5. It's nothing but a matrix where you indicate the rows are the species and the column is the reaction. So you have -1, -1, -1 for the first 3 species and 1 and 2 for these 2 species. So similarly, for the same first 3 steps of glycolysis that we saw, you can come up with the stoichiometric matrix and the equations. So for this, you have the stoichiometric matrix, which is this. Okay, so here you have 1, 2, 3, 4, 5, 6, because you have V1 to V6 reactions. But if you look at the number of columns, which is the number of uh, the substrate here in this case, or number of species that is involved, sorry, the column is the reactions, the rows are the species. What you see here, the last 2 is basically for ATP and ADP. These are also involved in the reaction, and what you see is ATP and ADP are involved in 3 reactions, one in V2, V7 and V4. Okay, so V2, V4 and V7. Okay, so this OD definitions can be calculated very easily. The sign indicates what is being produced, which part of the equation it's involved in, and or whether it's used up or whether it's being produced. And when you give initial parameters, initial concentrations and coefficients, then at a steady state concentration of, let's say, glucose at 0.357 molar, then or an atomic unit, or an AU here stands for an actual unit, meaning that it doesn't stand for a molar or a micromolar or millimolar. It's an arbitrary unit, so actual unit. So you see that the ATP increases and then it plateaus up. Glucose 6 phosphate starts getting produced, and fructose 6 phosphate starts getting produced. The same thing, if you flip it in terms of ATP and ADP, from the perspective of ATP and ADP, you can see what is being used, and how much of ATP is being produced and how much of ADP is being produced or used up. So these kinetic parameters, okay, here in this case, all these values, you can say that some of them are from experiments and some of them are arbitrary, like one, right? These are hard to obtain, and it depends on what type of experiments you're running, what experimental data is there to basically fill your equations with. And it's always a big challenge to estimate all the missing parameters. So in most um, systems biology ODE based models, you will see that uh, less than like around 30% of the equations will have experimental values, and rest of them will have arbitrary values given. Okay, either the concentration is at an astronomically high unit, or it's at zero, or something like that. Because the question in most of those models is not about what is the yield. The question in most of the models is about what is happening to the system, uh, like at a time point of, let's say, 2 minutes from the reaction starts, or 5 minutes from the reaction starts. Then there are, so ODE based models are not the only way in which you can do systems biology. There are many other models also. There is object oriented models, rule based models, constraint based models. This is a continuation of an ODE based model, but it's more used for an analysis called as flux based, flux balance analysis. I'm not sure if you have heard about it. Basically, it has a very high application in metabolic engineering and strain development in biotechnology industry. So constraint based models, flux balance analysis is a type of a constraint based model. Then there is interacting state machines, process algebra, which is a purely mathematical model. Then there is cellular automata, then agent-based systems, Bayesian networks, Boolean networks, where you use Boolean gates to check what is happening to the different entities as a with respect to time. So time component is there in all of these different models. So here is an overview of each type of model and what all can be seen or can be derived from each of these models. Like, for example, can you visualize Boolean networks? Yes. But can you visualize constraint based models? No, you cannot do that because it's a purely mathematically derived equation. It's very hard to visualize them. But at the same time, can you multi-scale or involve multi-state or compartments in a Boolean network? No. But you can do that with a rule-based model. You can do that with the with agent-based models. So this table actually tells you, in a way, what you can do, what you cannot do with different types of models that can be used in systems biology. It also tells you how you can, or if you have a question in mind, uh, what type of model would be best to answer that question. So you can use this as a general rule of thumb. Of course, in practice, systems biologists or computational biologists, what they do is that they make multiple models for the same question, and then they see which model answers them. So they either go one after the other, or they run all of them at the same time and then see whether a particular model is answering their question. The most okay, I'm out of time. I'm gonna uh, directly skip to this slide. Okay, so this is a an important thing to understand. I mean, if you are going to do systems biology, you should understand that the path is very important, or sometimes it determines what you're asking. So let's say there is S1 and S2 getting converted to P1 and P2, and there is a flow of information in this level, and at the same time S1 prime and P2 get converted to P1 prime and S2, and S2 gets used up again. When you the bread and butter of biochemistry has been carbon mass conservation. So when you look at equations, the balancing of equations, even for the glycolysis part that we did, we showed the stoichiometric matrix is basically you balance the carbons. Okay, 2 carbons this side got used up and 2 carbons got produced. Theoretically, you can go from S1 to P1 prime using this path. But when you look at carbon conservation, or from the point of view of carbon, this absolutely does not make any sense. Okay, it cannot happen. So it's a very invalid route of S1 to P2 and P1 prime. But this is a very valid route for finding out the perturbation that's happening for a system when, let us say, you have some component changing in S1, what is happening to P1 prime. So this is what something that throws up or comes out of many such kind of computational modeling, because these things are cannot be performed in experiments because it's invalid, but you can still come up with observations that have a that possibly change some of the paradigms. Is that all? May not be so, because you have a lot of other multi-substrate, multi-product networks, and these secondary metabolites complicate the picture. What I was going to show you is this. Usually the glycolysis pathway that we know from textbook is something that is like one after the other, like 10 steps in one. But actually, it's more complicated than that. What you see the textbook is this thick black line here, and what you see around it is also the metabolites and other accessory enzymes that are involved in glycolysis. So in reality, this is much closer to what glycolysis looks like rather than the textbook example of what glycolysis is. So there are different computer readable formats in which you can define a network, define a model. These are systems biology, ontology, cell ML. ML stands for markup language. There is SBML, which is widely used, the systems biology markup language. Then there are different databases. Most of you are familiar with this. KEGG, Gramine, which is for plant, grass plants. Then there is BioPsych. So the one of the if you want to start with some models that have been curated, tested, and works, you can go to biomodel database and you can download any one of the curated models, and these are they tell you whether it's ODE based or Boolean based or what it is. BioPsych website looks like this, where it has about 18,000 pathway genome databases from different organisms. Then tools, one of them you know already. In the next hands-on, we will go with Cytoscape, and we will see some of the things that we can do with Cytoscape and with networks. Okay, thank you. I know I skipped few slides, but you feel free to ask questions. Soundarya, some of the participants are not allowed to talk. Can you Make them talk. Any questions? Any doubts regarding biological networks or systems biology in general? Ankita. It doesn't show up here. Okay. Oh, okay. Thanks. Okay. Looks like no one has any questions. Either so either it all went inside or tangent. I don't know. Anyway, we will continue. Linux. Okay. So we'll take a short break. We'll rejoin at 11, and please connect using your Linux ID. Okay, not Windows, Linux IDs, and I'll see you guys. Myself and Dr. Krishnakant will see you guys at 11 o'clock.